So we have Guan. She's a PhD student in Nanjing University, and now she's in China. She's now visiting Japan, and she has published this really nice paper in PNAS some weeks ago as a part of her PhD in collaboration with the group of Peter Wallines. That's how I got aware of it. And this is a very nice work where they are trying to predict protein conformational motions using energetic frustration analysis and AlphaFold2. So, Jinwei, whenever you want, you are free to start.
Okay, should we start? Okay, greetings everyone. It's a great pleasure for me to be here and share with you our story, predicting protein conformational motions using energetic frustration analysis and AlphaFold2. From the title, you can see that it is about the intersection of how we guide AlphaFold2 with the concept of frustration from protein energy landscape theory, and how they together can offer us new insights into dynamics prediction of proteins. Now, let us start with AlphaFold2. AlphaFold2 represents a major triumph of neural networks and artificial intelligence. By leveraging the attention mechanism and deep transformer architectures, it predicts native structure from protein sequences with remarkable accuracy, almost on par with experimental methods. So, it is a great breakthrough in solving this difficult and important structure prediction problem and from merely sequence information. And according to the reasoning in Anfinsen's dogma, sequence determines structure and ultimately function. Then can we know about protein functions? Actually, only structures are not enough because we are missing something. That is the dynamics. Dynamics is important, but still, we don't have the same machine in predicting protein dynamics from only sequence information. And dynamics is of crucial importance because a lot of protein rely on their dynamics to function, to fulfill their functions, like catalysis, transportation, and signal transduction. Enzymes are the typical examples of efficiently and actively changing their conformations in the catalysis. And we know that enzyme function is the foundation of life itself. As we often say, life is movement, or in Spanish, la vida es movimiento. So dynamics is so important that with only dynamics, we can know more about protein functions. It's important and AlphaFold2 is so powerful. Then it is natural to ask, can AlphaFold2 also predict dynamics information from proteins? Anyway, it already works so well in predicting the native structure. That is the most optimal conformations, or the best solution. But unfortunately, this best solution is also the only solution. AlphaFold2 overlooks this dynamic information. Although dynamics is so important for protein functions, disease mechanisms, and protein design, but AlphaFold2 is so precise, or it is too precise, that it flattens out this dynamic information. And therefore, bringing this hidden dynamic information back to light is crucially important. As a result, many researchers have aimed to achieve this goal, to prompt AlphaFold2 to yield conformational diversity. And to understand such works, we have to take a further look into the frameworks of AlphaFold2. Suppose we have a sequence fed into AlphaFold2. Then the first thing it does is to collect a multiple sequence alignment around this query sequence, forming a set of similar sequences from various species. The idea here is that sequence correlations in coevolution often indicates spatial proximity. For example, if two residues always mutate together in the MSA space, then it is reasonable for us to infer that they are likely close together in 3D space and may even form contacts. With this reasoning in mind, we can say that AlphaFold2 looks like a translator. It translates sequence correlations into spatial proximity. And to explore conformational diversity within AlphaFold2, those works have a typical approach of involving reduced MSA. And they just reduced MSA to guide AlphaFold2 in generating alternative structures. I just mentioned that AlphaFold2 is a translator. It translates the sequence correlation within coevolution into spatial proximity. And by default, AlphaFold2 uses the entire MSA, essentially providing an average information. So it is pretty natural to see that in these works, when they use only shallow MSA or reduced MSA, because there are so many sequences in this MSA space, then shallow MSA can find those MSA variation, because different groups of them may carry different correlation patterns. Yes, AlphaFold2 is a translator, but what we learned from this work is that AlphaFold2 is also a sensitive and responsive translator, because it can give us different structures based on different MSA information. Here we can say that AlphaFold2 MSA works as prompts. It is very similar when you use GPT, like you ask GPT with the same query question, but with different prompts. Like explain why the sky is blue to a 5-year-old child is different from a physics undergraduate. And this situation is very similar in AlphaFold2 with query sequence and MSA as prompts. Then what is the major problem? The major problem is that we don't know how to write these prompts to guide AlphaFold2 to give us protein dynamics. And because we don't have this prompt, we don't have the guides of these prompts, and we lack this grammar book in writing this prompt, then we can only rely on random sampling. So we don't know which subset of MSA would give us which corresponding structures. Thus, it may yield unwanted structures or even unfolded structures by some subsets. What we need is a mapping rule. But this is very difficult because in the kingdom of AlphaFold2, the languages are first amino acids and then even high-dimensional tensor in neural networks. And remember, we are humans, so we cannot understand what do they really mean. So we cannot get this grammar book. But now, let us jump out of this data-driven kingdom for a while and remember that we have a biophysics grammar book, which summarized the principles of folding and dynamics in the perspective of biophysics. And if we use this grammar book as prompts to guide AlphaFold2, maybe we can get what we want. And this grammar book is Protein Energy Landscape Theory. Evolution gives protein energy landscapes a shape of funnel. So with an unfolded structure, it can slide in this funnel and fold itself correctly and efficiently. So the native structure is at the bottom with the most optimized energy. But things cannot always be so perfect. Let us look at this triangle system where this is antiferromagnetism system. We can see that in this antiferromagnetism system, the spins tend to adopt opposite orientation with its neighbors. And in this triangle, it is impossible for everyone to stay in the most comfortable situation. So this is frustration. Such frustration as external kinks and even extra basing to this funnel. And so we can have this protein energy landscape minimally frustrated globally, but it can still have locally highly frustrated residues. So is it something bad? Actually, this is important and favorable for dynamics, as we can imagine in this triangle system, because there is no comfortable situation, so the spins would always tend to turn up and down frequently. And also, protein utilizes this frustrated strategy to effectively changing its conformations. And this frustrated dynamics shown in this previous work is closely related to many crucial functions. Okay, now let us welcome our main character in protein, that is the ADK protein of this work. It has two states, that is the closed conformation and open conformation, where this closed state is the one and only prediction by the default AlphaFold2. And it has three domains, core domain, lead domain, and also NMP domain. It goes with large conformational change in its catalysis, turning this ATP and AMP into two ADPs. And in fact, it's so smart that it utilizes high frustration for an active mechanism of product release. Use this frustration to get over the speed bottleneck. And we used pretty awesome calculating tools to get a frustration profile of ADK protein. And thanks to these great works on calculation of frustration, because without any of these works, then I definitely couldn't be here presenting this story today. And I marked those highly frustrated contacts in red and minimally frustrated contacts in green. And from the figure on the left, we can already see that those red contacts, highly frustrated contacts, are already correlated with this conformational changing. And all those contacts that would break is called specific contacts. From the statistics, we can see that those highly frustrated contacts tend to be specific and involved in conformational changing. And this is also consistent with previously published work, where we can see that the pair of highly frustrated residues tend to be mobile. And this is also consistent with a large statistics in an illustrative set to show that this is actually a common rule. And I also have another variable shown here with the residue color from red to green. This is calculated by the mobility during conformational change. Red residues means that these residues have high mobility during conformational change, while green ones have low mobility. And again, from this figure, you can see that highly frustrated sites tend to be mobile during conformational change. So I listed many examples and many variables, and all of these are saying that frustration correlates with conformational change. And we're going to use it for our strategy. Okay, now let us forget about the open state of ADK. Suppose we don't know about the alternative state, and we only have this native state. And this is from AlphaFold2. And also, we have this MSS space. And also, we did the calculation of frustration of this closed state, which is also the native state. Suppose now we have the entire space of MSA prompts. And if we can only choose some of them, which perturbs those highly frustrated contacts or highly frustrated residues, then maybe we can efficiently break these contacts and push this structure out of its native basin from closed state to open state. So what we are going to do is that we will increase the instability onto those highly frustrated sites. Then how do we measure this instability? Actually, we measure them by energy profiles. So we map this MSA space, MSA prompts into energy profiles, and we focus on the energy difference between the query sequence with each from the MSA prompts. And we know that energy is a function for both sequence and structure. But we know that in the MSA space, all the sequences are still similar to the query sequence. So here, we do something like Taylor expansion. And we just keep this structure term unchanged. We keep it as the native state from the AlphaFold2 prediction and only modify the sequences. So what we do is that we thread every single sequence in MSA prompts and thread it on the native structure predicted by AlphaFold2. And then we just recalculate the energy by Rosetta. And because our aim is that we want to introduce instability onto those highly frustrated sites. So we only care about two measurements. One is the energy change on those highly frustrated sites, and also the total energy change. By such a strategy, we actually projected this MSA space using threading and Rosetta energy calculation into this frustration and energy space. Okay, and let us look at this frustration and energy space. If we have a similar range of total energy change, and we focus on the different instability introduced on those highly frustrated sites, we can see that in the left region, everything is stable. So we guess this will still yield the native state, that is the closed conformation. But on the right, though the total energy change is still similar, but now we can see those instability is added upon those highly frustrated sites. So we guess this will help generate those alternative structures, which is the open state. And we fed these sequences into AlphaFold2 and find that this aligns exactly well with our reasoning. And this is pretty robust because when we do random sampling in these two regions, we can get a distribution highly biased to closed states and open states respectively. And if we go further, we can get something like a phase diagram with a clear border between closed states and open states. And this is also consistent with our frustration and energy reasoning. And also we transferred our strategy to other proteins, and especially for this protein Chi-B, which is a typical example in AF cluster, because it involves highly complicated changes in the secondary structure in a fold switch pattern. But we can still successfully get something similar with protein ADK. Okay, and again, we can show that our methods of perturbating highly frustrated sites also aligns with breaking specific contacts. As I showed you earlier, I talked about how high frustration is correlated with contact breakage. So now, suppose that we have the real answer of the open state that is taken from the PDB bank as a crystal structure. Then for each MSA prompt sequence, because we know that which contacts are shared and which contacts are specific that would break and form during this conformational change, then we can define this order parameter. This is called Z-gap. This shows how prominent is the energy of specific contacts compared with those shared contacts. And we can see if the gap is high, then specific contacts have prominent energy, so it would be breaking or tend to break. And it aligns well with our structure prediction upon this frustration and energy approach. And also we have this distribution with those structures predicted to be closed and open have differences in the distribution of the Z-gap. Okay, now we use the various methods and we successfully predicted the alternative structure with AlphaFold2. And also, we made the mapping rule between sequence correlation and spatial correlation in AlphaFold2 much more understandable, because our method is based on biophysical logic. And also the same strategy may be generalized for other proteins with large conformational change. But now we just got two distinct states. And it would be more valuable if we can get a conformational changing pathway between them. And we just find two methods. The one is that we can introduce this instability progressively by selecting the MSA step by step from left to the right. And now we have created a continuous pathway from closed state to open state. And the second method is equally intuitive, where we just do interpolation using random mixing. We have the most stable set and the most unstable set, and just randomly mix them. It also generates a continuous transition pathway. And also because ADK has three domains, then we can use the angles in between to characterize its conformational change. So we projected our results onto this angular plane, and we can see that here is a continuous changing path, and this path correlates well with experiments, and also the all-atom simulation if you look at those gray spots. And we still have another method of masking highly frustrated sites to introduce the instability onto those highly frustrated sites. And this is inspired by this work where they mutate residues in this MSA space. Because when performing multiple sequence alignment, if the lengths of sequences differ, then empty spaces would be added to fill the gaps. So if we artificially masking the residues by substituting them with this empty space. Actually, we are doing something like breaking the contacts, because I showed you that coevolution means spatial proximity and means contacts. So when we randomly masking the residues on those highly frustrated residues, we can see that we are pushing the structure out of its native basin. And this is highly susceptible, because we can see that protein ADK has a sequence more than 200 residues, but mutating the top 10 or top 30 by masking can push this conformational change. Again, this is highly non-trivial, because we can see that we do the same thing on highly frustrated sites, or those residues are rarely correlated with conformational change, then it biases this distribution towards the alternative state. But if we do the same on those minimally frustrated sites or randomly chosen sites, then we won't have the same effect. Okay, now we get alternative structures and also conformational changing pathways. We use the various detailed methods, so our reasoning is quite robust. And can we push these insights even further into areas like protein design? Okay, let's say, because we have the conformational changing pathway generated for ADK, we can see that ADK first open its lead domain and then an MP domain. So this conformation is quite common, while the opposite way, with an open NMP and closed lead domain, is really rare. It's already very rare with filtered MSA prompts. And also, the entire MSA space can only generate the default version, that is, with both domains closed. So how can we do something to generate such conformations? And because I said that we use the energy profiles to do the modification in MSS space, and AlphaFold2 can translate and follows our reasoning. So in MSS space, we just destabilize NMP domain and stabilize the lead domain by some mutations. But remember, we didn't do real mutation on those query sequence, and we only modify in MSS space to create a more stable lead domain and more unstable NMP domain. And what did we get? And we get this rare conformation with an open NMP domain and lead domain. And please pay attention that this is not about the entire state of this protein ADK, because now we are mutating or we are changing the behavior of distinct domains. So we can do it really fine and into distinct domains. This shows that our reasoning may be a possible strategy for protein design. Okay, here is our summary. And what we did is that AlphaFold2 is a technique that turns sequence into structures. But we also have theoretical principles in biophysics that generally summarize the principles, like how would a sequence fold and how is its dynamics. And what we did is use this biophysical logic to guide AlphaFold2. And finally, we can also get dynamic out of AlphaFold2. So we offer new insights of combining energy landscape theory with data-driven approach. And we provide a possible new strategy based on biophysics for dynamics prediction. But also, we have some limitations. The first thing is that our throughput is rather limited because we have to calculate frustration and energy. But actually, now we are working on a new method, and I hope I still can have the chance in the future to share it with you. And also, conformational change. The scale of a conformational change is also a problem, because we did the proteins with large conformational change, but it would be less effective for those small-scale motions. And also, in our methods, the accuracy is dependent on the estimation of energy and frustration. And the most limitation is that if a conformational change is strongly correlated with the ligand interaction, then maybe it could not be so effective, because in ligand we need the conformation and also the energy function. But I think in the future, AlphaFold3 can help us. Okay, I think this is the end of my presentation, and I sincerely thank our team members of this work, and I thank for your organization. and invitation.